Okay. Good morning, everybody. Good morning. We are starting. I'm Ville Merta. I'm the president of the FlowTex. On the behalf of our organizing committee, welcome everybody. We actually started already yesterday with the best practices, more educational day. Today, we start with clinical session, and then we continue with more or scientific research talks later. It's been really great to see we are kind of growing every year a little bit. We have this year more than 300 in-person registered attendance and similar or even higher number of online viewers. So we start with clinical session, and Marsha is our lead on that. She's been or came part of the FlowTex on her days in Baylor College of Medicine and Methodist Research Institute, and then later moved to the industry where she has really been working more on the clinical side. Please, Marsha.
Thank you and good morning. So I guess all of you can blame David Haviland for getting me interested and involved in flow cytometry, and probably also Dorothy Lewis over at Baylor College of Medicine. I think it's just so precious that we actually have this meeting every year, and we have a clinical session right next to a research session in the heart of the Houston Medical Center, because so much of what's done in the clinical lab comes from the research that's done by research scientists right here in the Med Center and worldwide. And clinicians really rely on those papers that come out, and they get in their labs and put together tests that can change outcomes for patients. And so I'm especially excited about today's presentations because they follow a linear path. We're going to talk a little bit about what it takes to set up the most recent instrument. When the clinical version of FlowTex started, a lot of people were still running on Caliber and the first Canto 2. We've since transitioned from 10-color Canto all the way into the Lyric from a BD perspective, right? There's been certainly Gallios and Navios from Coulter and more instruments coming. But I think the reason that I'm bringing all of that up is we couldn't be here today having this very important conversation if we hadn't come together across Texas and then eventually across the United States all those years ago. And so really excited to hear from our speakers. Very honored to have also Virginia Litwin, who I'll introduce later, our ISAC president-elect. But first and foremost, Jolene is going to kick it off for us. And it's my distinct pleasure to have her here because as we were forming, we played on the word FlowTex and we called it text flow when we came out with the clinical standardization and put together the first 10-color tubes, very much working with UT Southwestern and MD Anderson and labs really all across Texas. But we were able to bring in the JoJo crowd, which is Joe DiGiuseppe and then Jolene, who's here to speak. And she's really, we say the clinical version of FlowTex has been around for about 13 years now, and Jolene has been working at Hartford Hospital for over 28 years. She leads CAP investigations. She also leads CAP training. She's really dedicated her life to the advancement of flow cytometry, and flow cytometry is really fortunate to have somebody who thinks so innovatively at the same time that can put the quality control behind it to protect the patient. And so she did put together the very first 12-color tube that was ever shown on the BD Fax Lyric. It was a B-cell tube, and we showed it at the ICCS conference in 2019. And so no one, I don't think, better equipped to come and speak to us today, and then just really honored because of what she's already invested personally in this group. So with that, Jolene, I will give you the floor.
Thank you, Marsha, for those kind words. All right, good morning, everybody, and thank you again, Marcia, for those kind words, and thank you for TextFlow for having me here. I'm going to be talking about instrument validation, and I'm lovingly calling this my 7 stages of grief because in my 28 years, I've had a couple instrument validations, and this was none like the other, and I'll tell you my story. I have to click one. Clicking. Oh, here we go. So the first stage of grief is called a shock or getting ready. And in our lab, gone through a couple of validations. So it's the same which instrument you have, just getting ready to where to put these new flow cytometers. The footprint was a little bit smaller than our 10 color Cantos, but nonetheless, we had to find some counter space to put these flow cytometers. We actually went vertically. We had the lyric, and then we put the monitors on top of it to sort of maximize our space there. Also, prepare for some new considerations. So at that time, we didn't have these UPSs, the uninterruptible power supply. And we thought in Connecticut, we do have some blizzards and some brownouts. So maybe we'll get these UPSs in when we get the lyrics in. And they also, as an added bonus, helped mute some electronic noise, which was big for the lyric with that background noise. And then prepare for validation. So we had our game plan from before, but revisiting and go through that. And I'll go through that in a minute. And some reagent supply. So at the 10 color Canto, we did have CS&T beads, but the fax lyric came with its own set of CS&T beads. So we had to make sure get those in stock and the comp beads that were needed for the fax lyric or those FC beads. And then thinking about the instrument setup. So getting the voltages ready for each assay. So our standard panels, leukemias, lymphomas, our PNH. Yes, we're looking at red cells. So looking at optimizing our voltages for the size of a red cell and for our assays that were permeabilized. So thinking about those voltages. And then our compensation controls, as we said, looking at the FC beads that we needed for that. And then we're ready to go perform that validation. So let's look at the game plan that we have here at Harvard Hospital. So just talking about different things that we're going to evaluate, the accuracy, precision, all pretty specific from the CAP checklist, and then the process that we were going to go through, review the data and collect the data and how we are going to analyze the data. So we were ready. So bring it on. We're ready. But here is the work order that we had for this fax lyric coming in. And you can see here the date. This is April 2020. And there was absolutely nothing going on in the world at this time. So we were a little panicked there. So what did that mean at Harvard Hospital? So just a side note, this is our cafeteria, and we actually had toilet paper and paper towels for sale in our cafeteria because that was what you did back then. And here at Harvard Hospital, this is our receiving area, and this right here is a refrigerated morgue trailer. So we had so many deaths at the time during this pandemic that they actually had to have a refrigerated morgue trailer. But this is where our truck was coming in with our lyric, right next to this morgue trailer. So they delivered it, brought the box in. Now, no visitors could come into the hospital, so we had to go get this container and bring it into the lab and start unpacking it, putting it on our counter. And then the field service engineers came in. This is not them, but they came in like this in hazmat suits. It's totally separate from any lab staff. They did their thing and they got out. And usually during some instrument installs, there's some back and forth with the engineer. What are you doing? What are you looking at? What are you setting up? And there was none of this interaction. There was a sort of a missing piece here. And then also for training, since there was no travel, we could not go out to California, go to BD school and learn, the nuts and bolts of the operation, but we had FaceTime. So we did a lot of our training through FaceTime, had an excellent application scientist who walked me through a lot of things through FaceTime and helped get us up to go. And at Harvard Hospital, this was a staff. We had to maintain 6 feet of spacing. So training was sort of an issue too, but we were ready. So for the love of God, just let this go okay. So the first piece, looking at accuracy, so pretty standard with any validation, getting our 20 samples, abnormal and normal for these quantitative assays. And we looking at the 10 color lyric versus the 10 color Canto versus the lyric, and how we were going to review the data is just put it into an Excel program and look at some statistics. It's going to take a sip here while you look at my graph. So here, I'm just going to isolate and look at the 3% old and new, and the old in red and the new in blue, and the 4% old in red and new in blue. And I'm going to plot here in Excel, and you can see my R-square looks pretty good for all of these. The results are pretty concordant. Prepare this report. Our acceptable limits were greater than 0.95. And the director, my other JoJo here, signed off on this report. And then looking at our qualitative assays for accuracy, getting our 20 samples, normal and abnormal, comparing again the Canto and Lyric. And we were going to look to see if the panels were similar and would we get the same diagnosis. So here, looking at our old cytometer, what was positive and negative for our cases, and hopefully on our new cytometer, we had positive and negative, thus similar diagnosis, and our accuracy and our specificity and sensitivity then could be calculated. And our criteria was hopefully that we got 95% or greater on those values. And we wanted to make sure we had each sample type. So in my lab, we call them bloodies and clears. So we actually stain, lyse, and then wash off the unbound antibody. So since we're having that stain lyse option, so we have our bloods and our bone marrow. So we wanted to make sure we had enough normals and abnormals of our bloody things. And we have our clear specimens, or our fluids, our lymph nodes, our CSFs and whatnot. So we want to make sure we had enough 20 of those are abnormals and normals. So here is our leukemia and lymphoma PNH testing that we were concordance with all of our panels. And our leukemias and lymphomas, that was pretty easy to get. Now PNH we know is a rare disease, so this was kind of a hard nugget to go after. So we do have a patient in Connecticut who we have been monitoring, but now is being treated, so we don't see this person anymore, but we asked for a specimen to come into the lab, and we serially diluted, got 3 data points from that patient, and then using the CAP samples, I was able to get some more positive specimens, of course enough negative specimens, but so to satisfy the PNH abnormal component there. And then precision, that can be split into 2 ways. The first way is intra-assay, so samples within the same run. And looking at the 2 assays that I have, the CD4 and the TBNK, using my normal and low controls, I set up that sample and acquired 5 to 10 times until the sample was exhausted, and then looking at some statistics here. And you can see here is my normal run 5 times, and then in green, the statistical analysis for those. And then here below my low that was run 5 times, and again in green, my statistical analysis for that. And again, for intra-assay, now samples within the same run, again, looking at our qualitative panels, so our leukemias and lymphomas, looking at our normal and abnormal specimens, setting them up in duplicate by 2 different operators just for ease of use there, or workload, and we acquired them in the same run, and just looking to making sure that we got the same diagnosis. So here are our samples, our different runs by the different operators, and we're making sure that the final diagnosis would be the same across all of these runs and specimens, and we got concordance here. The second component for precision is inter-assay, so between runs. So again, looking at the CD4 and TBNK, those normal and low controls, setting them up in triplicate and running them at different time points, and then looking at statistics again. So here is our normal specimen. Here in yellow, looking at this normal 1, 2, and 3 run around 7 p.m. here. Our normal in blue, our normal 1, 2, and 3 run just before 5 o'clock the next day. And then here in orange, again, our normal 1, 2, and 3 run a little bit after 8 that same day. And getting all these different time points, and then again here in green, looking at our statistical analysis. For qualitative assays for between run, again, looking at normals and abnormals and at different time points, and would you get the same diagnosis? And here are our samples, ran them in the beginning and making sure I had enough specimen to run at these different time points and getting the same diagnosis. And this is sort of real life here, because you might have the sample set up and ready to run, but the analyzer might not be ready to run at that time. So we're putting it into the refrigerator, ready to run later. So this is kind of a real world scenario, making sure whatever time we run it at, we will get the same diagnosis. The next phase in validation was carryover. And really, this pertains to any flow cytometer or any automated pipetting system. So any processing assistance, they are delivering a precise volume of blood sometimes. So if the probe is changed or replaced, check the guidelines for those probes that it's delivering the proper amount of specimen and reagent. And for the flow cytometers, so for our lyric, we set up one sample manually rather than putting it on our processing assistant, our lice watch assistant, and adding that variable in. We set it up manually, followed by 5 PBS tubes, and then checking the limits of carryover for our Defects lyric. Here for the sample carryover with the default 1 wash, the carryover should be less than 0.1%. And for 3 washes, for that probe, 3 washes, the carryover was had to be less than 0.05%. So here is our carrier assessment that we did at Hartford Hospital, and you can see again the acceptable limits, less than 0.1 and less than 0.05. And our fully stained tube, followed by the unstained PBS tubes. And I looked at what I would consider an actual viable event. I didn't want to account for a speck of dust that might be in there and not be counted as a cell anyway. So I wanted to look at what I would consider a viable event. And I was indeed less than the acceptable limit of 0.1. So this was acceptable for that 1 probe wash. Now, what if I did a couple more washes? 3 washes of that probe, the stained tube, again, with 3 washes of that probe, and then it would go into this PBS tube, the 5 PBS tubes. And what I would consider a viable event, now my acceptable limit is pretty good here. I'm less than that 0.05. I am less than that, but what about MRD? Is that low enough? So at Hartford Hospital, we go down to 0.01. So this would be a little bit kind of worrisome for MRD analysis. So there is a checklist requirement that says if you do have some carryover, you're putting some steps into place. So in my laboratory, not only on the lice wash assistant, but also on the fax lyric, we put water tubes in between our fully stained tubes. So if there's any amount of carryover, it will go into that water tube before it goes into the next stained tube. We also, so that would be for our rare event analysis, our MRD and our PNH assays. And we also do a unstained tube where we don't set up any antibodies on this sample except CD45 for gating purposes. We run that unstained tube to make sure that all the parameters are, that there's no carryover in that unstained tube. If there is some carryover, we would see it in those channels. And we also use that unstained tube for autofluorescence. So we can see where the positive and negative begins and ends, and that's can come into play with APMLs to see where the positive and negative begins and ends. So next part of validation, looking at linearity or the analytical measurement range, the AMR. So you have to check your instrument, your assay, and your specific ranges for that assay, and serially dilute to cover the AMR of that assay. So you can see, you can test it goes as low as it's advertised to go, and it go as high as the range here. So serially diluting, we chose a pediatric patient to make sure we can go as high as we could go, and diluting it down to show that we can get as low as here. Now you cannot report above or below these reference ranges. So for example, this subset here, I can only report down low as 5. And if my patient was 4, well, I couldn't report that. I would have to report less than 5 for that patient. So we completed our validation. We wrote this 12-page report, signed off on that, ready to go and launch. But then we entered our second phase, a denial, what didn't work as expected. So we had approximately 20 cocktails previously validated on that 10-color Canto, 8-color mostly, some 10-color cocktails, and they had been in use for 10 to 15 years. But what we were finding as we started to look at our panels and our tubes, that some didn't look appropriately compensated. So we started troubleshooting for possible errors. And here in the next slides, I'm going to get kind of lyric specific, going through different ways to look at your spillover values and different tips and tricks that we learn over the time to different methods. But I also go through some basic compensation strategies and concepts like comp controls and how to evaluate for compensation. So here, how to look for good comps and bad comps. So we know, looking at it, compensation just right, the positive is centered right over the negative. So this would be good compensation here. Over on the left, this would be over comped, and on the right, this would be under comp. So we know how to look for compensation using, this is a bi-exponential display here. So we can see what's above and below the zero. And this was one of my tubes that we can see that we have some overcompensation trouble spots here. This DR BV605 was overcomped, and the CD19 APC R700 was overcomped. So then we entered another pain point, this anger phase, where like, what are we going to do? We went through the appropriate plan. We did our FC beads, and we got our spillover matrix all calculated. But this is I'm going to show you, this is a table from a module that we put out through ICCS. And this is going to help us look at those spillover values and what we can do with that. And by compensation matrix, I'm referring to this spillover values that's in the facts lyric, looking at those 12 versus 12, that was the compensation value set by the FC beads. And this option 1 is going to look at some trouble spots and what I can do by choosing my own compensation controls and replacing what used to be set by the FC bead with my own compensation control. So first, to do this, I need to go to my library under reagents and actually add those two into the library here. So I'm adding the CD19 APC R700 to my library, and I'm choosing the lot ID as specific. I can actually put in the lot ID from the vial, but rather than chasing lot after lot, I just put the word specific, and then I choose a date far ahead in the future. And now I'm ready to run those reference settings. So here, I'm going to right click on my PB new tube 6 I have, for example, here. I'm going to right click on that and choose this create reference settings option. And I'm going to program those two in, so that BV605 and the R700. I'm going to choose my control type that's linked to cells. I knew those to work back in the 10 color days. I was more used to cell-based comp controls. And I'm going to put in my labels that I've already programmed into my library. And I'm going to choose that lot specific one that I programmed in. And I'm going to choose next, so I can go forth and launch the program here using the CSNT bead first. And I've already set that here, which lot I'm going to use. And then I'm ready to run these two cell-based comp controls, single stain comp controls. And in here, I didn't have a negative or an unstained because I'm gating around my lymphocytes, and that will capture a negative and a positive population all together. And when you're running these, make sure that they are lymph-based or lymph-gated or monocyte-gated. So for instance, if you're running CD19 and CD8, you can gate around your lymphocytes and capture some positive and negative populations in that lymph gate for the CD19 and the CD8. And for, say, monocyte gated, so say if you're running CD33 or CD14, you're able to gate around the monocytes and get some positive and negative there. So now with this method, I've optimized the compensation for one tube with my preferred comp controls, so cell-based comp controls. I do not have a 60-day out date anymore because they're not entirely based on the FC beads anymore. And anytime I get a new tandem in that tube for that R700, the BV605, I can easily update my reference settings for that tube. But what if I want to do that for all antibodies, for all my tubes? Well, here it gets pretty complicated. And I did have to phone a friend. I reached out to my TSA, Maureen, who awesomely helped me again on FaceTime setting this up. So for option 2 here, I'm going to entirely replace my lice wash reference settings. And this can be cumbersome, as I said, to set up. And that's why I phoned my friend to help me get this set up. So just to revisit what comp controls I was going to run for these, I took my data that I had from the 10 color fax Canto. And for this fluorochrome, beads seemed to be better. They compensated better with the CD64 R700. And here, using cells seemed to be better in this panel using CD7BV421. So using these comp controls, and I was ready to get the reference settings set for my whole lice wash reference settings, this whole matrix to be replaced with my preferred comp controls. So to do that under the library in BD, under the lice wash, I went to that spillover tabs. And then over here, you have this remove fluorochrome option. And don't worry, by clicking this, you're not actually going to remove anything yet, but this screen will launch. And here you're able to select the fluorochromes that you want to remove. And the trick here is to remove everything except one. So I went through and I clicked everything except FTSE. And then I took a screenshot of it to remember which ones I deleted and I needed to put back in eventually. So once I deleted, clicked all these off, then I clicked remove. And it's going to remove those now from your lice wash reference settings. And then now I need to add them back in. So I'm going to add those fluorochromes to my lice wash. Again, remembering if I'm adding a tandem, I have to first add that to the library. So I'm able to choose that like I did that CD19 ABCR 700, choose my cell-based comp controls, my lot ID specific, and run all of those comp controls, remembering the lymph ones runs with lymphs and the monogated running with monos. And then after all those were put back into the lice wash reference settings, then I removed the FTSE like I did before and then added the FTSE in this way. So now I have my new spillover values entirely with no expiration date. So no 60 days here at all. And this is now chosen by me when I want to redo my spillover values. So anytime anything in our optical pathway or lasers or whatever is changed out in our optical pathway, that's when we will reset these spillover values. So how did that work for me? So before, as we saw, there was some overcompensation errors. And now I seem to have fixed a couple, but not all of them. And one actually changed a whole different way. So a little sad, this flipped our lab upside down a little bit because now I did the recommended way, I did my own way, and nothing seemed to work here. This is actually last summer, my daughter and I went on this whitewater rafting trip up in Maine, and this is her falling out, and this is me down here in the water. It was fun at the time, but a little stressing, just like it was for us with our 7 stages of grief. But so our lab got flipped up, but you know, we were gonna power through and start reaching out through help. So we did reach out to the vendor, working through some of the specifics maybe we missed in our FaceTime training, but they couldn't really recommend anything with our panels, our lab developed tests, as you know, they couldn't really do that. So we started to meet regularly with them as well as some colleagues. So we weren't traveling to conferences at the time, but still on the phone for meetings through Zoom. So, oh, you have the lyric. Oh, you have the lyric. Oh, what are you doing? So we started to all reach out together to work together on how we can share some ideas, tips and tricks, and working with the vendor. We sort of tightened up some of our practice. So one of them was the CS&T beads. On the 10 color canto, we could take them all around and spin them around all day and run it here, run it there. So on the lyrics, we would ran our CS&T on one lyric and then other lyric. And we said, you know, the product insert really says 20 minutes. So we write on the tube now when we make those CS&Ts and make sure we use them in 20 minutes. Another thing that we learned, tighten up some of our practices, was the BSB buffer. So that brilliant stain buffer is used to mitigate any brilliant stain dye interactions when they're put together in that fully stained tube, but we weren't putting them into our single stain comp controls because that didn't make sense, right? Like there's no other brilliant dye in there to interact, but you have to keep the conditions the same. So whatever you're putting in your fully stained tube, you're actually putting that into your single stain tube. So things that we learned as we went forward, but also some ideas and tips that the colleagues we worked through to brainstorm together. So here is one that Val Miller in University of Washington, working with Sindhu Cherian, she came up with this word trickery. So this is option 3. And actually do this in my lab in 2 scenarios. So scenario 1, where the antibody that is used is on a small population of cells. So CD38 on plasma cells or CD34 on blasts. I might not have enough events in there to acquire enough population of cells for the spillover values to be calculated. So I'm going to use trickery for that. And in scenario 2, where the spillover values just weren't correctly calculating to compensate for the populations. So here, what trickery means is you're staining a fresh normal blood. In this example, using CD8. CD8 is nice and bright. And using CD8 in the same fluorochrome that for the antibody that's in the lice wash. So an example, you would in the wizard select CD34 APC, where you're actually running a sample stained with CD8. So that would be. So here, this is Val Miller's data from the University of Washington. So I have to acknowledge her data here. Her first attempt to add CD56 AF48 to her lice wash. And it didn't look so pretty. There was a lot of overcompensation, which she actually used that CD56 antibody. So she decided to use her trickery by adding CD8. So she it has to add a whole new lot so she can use that trickery. So her lot ID, she actually uses the date that she added it to the library, which was 2/8 and her expiration date. So once she ran the CD8 instead of the CD56, well, first she had to add that to her do property, so it would actually grab that value after she ran that CD8 comp control. So now when she's ready to run the sample, it'll grab those spillover values. And her single stained tube here looks pretty good. It looks better than it did before. So reviewing this single stained sample here, you can see that it looks compensated, but really to test it out, test it on a fully stained tube. And this looked pretty good. Everything looked pretty perpendicular, no worry spots here. So now for her SOP, when she's adding CD56 AF48 into her lice wash, she's staining the blood with CD8 instead. And then before to, because she has that in a couple of her assays, she has to go in the library and add that to her assay for that newly approved lot. So adding that CD8 trickery to all of her assays that she's going to run here. And then that began stage 5 for us because it seemed a lot like this. I didn't have the bandwidth to go to my refrigerator and okay, CD8 worked for some, but it didn't work for all. So what in my refrigerator of antibodies might work here? A bright antibody, of course, you want to have as bright as what you're using for compensation. But I just didn't have the bandwidth to, you know, Val has a whole recipe card that she figured out what works for all of her panels and assays. So I just didn't have the bandwidth at the time. And we are still in the middle of the pandemics, you know, separating and 6 feet of separation and whatnot. So came across this last method, option 4, and this one gets really complicated. Again, phoning my friend Maureen, who helped me with this. So what this is your modified reference setting. So you're setting that you're running your compensation controls, but then you're able to edit them and save them into your spillover values. So here I have to give kudos to Bruce Gregg and Vanderbilt University, who has supplied this data for me. Another colleague of mine that helped work through our tips and tricks here. And you can see he's got some trouble spots with this APC. Here it is over compensated with the P size 7, and it's undercompensated with the APC H7. So in his spillover values here, looking at the tab under APC with those 2 parameters, we know that 3 is a little bit too much. So it's overcompensated here. And the H7, this 9 is not enough. I need to make this number a little bit bigger to make this better compensated. So once those values were toggled, the compensation looks a little bit better. And here are the values that I have now. So we knew 3 was too much, but 0.5 seems to be just right. And the H7 we knew was not enough. So we went from 9 to 14. So this is kind of like Goldilocks, a little bit too much, a little not enough. So we got it just about right here. So now that we have those values, how do we edit them into our spillover matrix value or table? So here, this is just a sample example. We have a tube called Jovi tube. I'm going to right click on that and save those modified settings. So I'm going to save those into my reference settings. And it used to say number 15 here, but now this is going to be my version 16. So I've created a new reference settings here now, and I'm going to finish. But I'm not quite done yet. So I got my reference settings for this tube, but now I have to replace it onto the assay. So when I run it on my work list manager, I'm able to grab those modified reference settings each time. So I'm just going to create and replace that existing assay here, replacing. But now I have multiple tubes with that. So I have to go in here each time and put the two properties to now number 16 for each of these assays. It doesn't take long, but still going through this, 5 or 10 minutes. I have 2 lyrics. So this is just one tube that I'm modifying over and over to get those different assays set up. In this Jovi tube, this is our T-cell tube where we have TRBC1 and the clone is Jovi. So it seemed like a cool name to name our tube Jovi. And we leave the PE channel empty so that we can actually drop these markers in for whatever clinical neoplasm that we want to detect here. So I'm picking each one of these and making sure now I'm choosing CD16 for those two settings. So now we're entering our phase of acceptance and hope. We seem to have a method that is going to work for us. So designing our new methods and new tricks, we designed some, in the process, we designed some new tubes, maybe some of our panels that were optimized on the Canto. We need to redesign now for our lyric. We also look at our cocktail stability. Before they were for 4 weeks, but now we thought that they would be more stable at 2 weeks by looking at our fully stained cocktail, setting up manually, looking at like how long we could carry that out until things got a little bit wonky. So 2 weeks seemed like a safe stability time for our cocktails. And then constant vigilance. So in this time period, we were doing post-acquisition analysis until we got our system into place. So here again, when every cocktail is made, we verify that the antibodies are added. So we've added our TRBC1. Again, this P channel, nobody forgot anything. We left this empty for our drop-in channel. And yes, CD16 was added, 56 was added, CD4, et cetera. So we're verifying that the cocktail was made correctly, but also we're looking at every X and Y of this tube. So we're making sure everything is properly compensated. So this is like 6 pages of worksheets looking at every possible X and Y combination. And I do this on when a cocktail is made to verify that the compensation is going to work for the lifetime of that cocktail, because there might be a new tandem lot that's in there. Now I can change it right for that cocktail, and it'll be good in the spillover values for the lifetime of that cocktail. So last stage, processing, moving through our accepting, so reliving the pain points with other colleagues. We've put out this module on the ICC website. We also made a webinar feeding back to the BD school on maybe things to emphasize that we couldn't grasp through FaceTime, maybe stress a little bit more in school. So working with BD a little bit. And so finally feeling like the system is under control. And we've had the lyrics now for 4 years, and I can't believe we're almost ready to get on our 5th year and think about that. And then again, my director and mentor, Joe Giuseppe, this is the JoJo team here, has lived with me through these 28 years at at Harvard Hospital. So help process these 7 stages of grief with me. And then my team here who helped get, this input set up correctly. So the output for Joe Giuseppe to make his clinical decisions. And with that, I'll take any questions. Okay, you've all lived through the 7 stages of grief. Thank you.
Were there any questions online? Not yet. So I would say don't go so quickly, right? I think one of the things that we're going to get into a little bit later this afternoon is what the FDA is considering to do with lab developed tests, which clearly your talk was all about lab developed tests. And I just would love for you to come up and speak just a little bit to the rigor, right? Like if you saw 16 iterations, like there was no, oh, we're just going to run this and see what happens. So what do you think? What do you think? One of the things driving that is patient risk, right? So the risk of getting a wrong diagnosis to a patient. Outside of the steps that you showed today, are there other tools that you guys use to avoid sending out a misdiagnosis?
Well, there's so many possibilities that technologists have to be savvy about. Carryover, contamination, looking at our positive and negative fluorescence, autofluorescence, bubbles in the system that you don't want to be misdiagnosed as something. So many pieces of validation, making sure your data, looks appropriate. When something goes wonky, it's like, well, okay, what is tricking me here? What happened here? So just troubleshooting, looking at the parameters over time. Was there a bubble in there? What did my unstained control tube look at? So thinking, is FDA going to look at all of that and be able to dive into that and make sure that the data is robust, that the laboratories are providing. So, just thinking through those steps.
Yeah. And then I think too, not that you're a clinician, but is this flow cytometry all that's used to diagnose a patient?
No, so there's only certain entities that can be entirely diagnosed by flow cytometry, CLL, hairy cell, and some, need molecular markers to either diagnose or give some prognosis. So, I mean, hopefully there are some, catches to the whole system, but it is taking the immunohistochemistry and the molecular cytogenetics to make that a diagnosis.
And then my very last question, and maybe Dr. Wong, you would chime in a little bit here, but I think this is a bit of a boot camp, because as we start to move into even more complex technologies, potentially, like if spectral comes into the clinical lab, there will be, I would say, an even bigger.
Yeah, I can't even think about spectrum. I'm just wrapping my brain around spillover values. So, there's so much in the future that to allow for innovation, and even immunotherapy, there's so much more markers being targeted now than when I started 28 years ago. So what's going to happen in the next 28 years, being able to diagnose and find those markers with immunotherapy that's coming out. So hopefully, everything can just keep on going.
One last, is there an average number of markers you guys use per patient?
Average markers. So it's a 12 color fax lyric. We haven't filled all 12 colors. We safely go with 11. So we use maybe we bill out right now, say an average of CD19, average number of 19 markers per patient. Yeah.
Well, thanks, Jolene. Okay. Very nice job. So moving right along, intentionally, I asked Virginia to come and talk to us a bit. So both Jolene and Virginia and others have contributed to ICCS putting together just guidelines, right? So clinical guidelines that have been published in Cytometry B and are used, I would say, by clinical labs across the United States. But even deeper than that, Virginia has played a pretty important role. She's been a part of NIST, which is National Institute of Standards. She's also participated in a writing of a recent guideline, HL, and I always butcher the H62 and H42. So those came from CLSI document development committees, and the FDA was actually part of writing both of those documents, and she's gonna, come up and talk about some of, I would say, the struggles we still have, even with all of the work that was put into the H62 guidelines. So she right now currently is the ISAC president-elect. She was also the 2023 ICCS Wallace H. Coulter Award winner. So you missed a great talk that she gave there, really overviewing her career and her journey, through NIH and other collaborations that she's had and mentors that she's had through her life. So I'm really actually so delighted to have her here with us today to help this important conversation, but really right now to lay the groundwork for what it looks like to have guidelines in flow cytometry. So Virginia, the floor is yours.
Yeah, we can use it. Use your mic.
Thank you, Marcia, for the really kind introduction. And it's a pleasure to be here. I mean, coming to FlowTex is on my bucket list. And so it's really a pleasure. And I also want to acknowledge Marcia's secret superpower, which is to bring together really good panels and bringing the right people together. This is not the first time I've collaborated with her on something. And each time I meet some interesting people that I keep in touch with, and you'll see that with our other speakers today. So I prepared this talk in light of the panel discussion is for addressing the FDA's proposal that they should regulate laboratory developed tests. And so we need to understand how we develop how we do flow cytometry validation, as well as how we would review those validations. And then how we would advance the slides. Okay. Okay, there we go. So when we talk about understanding flow cytometry validation, Marcia mentioned a couple documents that we put together on that topic. And the reason we did that is validation documents for other technologies are not fully applicable to flow cytometry. I don't have time today to go over, all the differences, but there's some unique challenges with flow cytometry. And what I'll talk about today, again, in light of the FDA proposal, is a few things that I've highlighted here in red. And so one is the lack of true reference standards for flow cytometry. And this gets into how can we do accuracy without that. And as Jolene already addressed, the rapid rate of technological advances. And when we're reviewing flow cytometry data, which is what the FDA wants to be doing, is we need to understand the technology. And I'll go over a little bit today, but how the validation terminology, it's got its own language, translates to flow cytometry, and understanding the context of use of our data. So this is the document Marcia mentioned. I'll be referring to it during the presentation, and it's the validation of assays performed by flow cytometry. In this document, we take a tiered approach to developing your validation strategy, and this is based on a lot of factors. One of the most important is the context of use of your data. We strongly want everybody, whether you're in a regulated lab or not, to be doing some validation, and we want to have this tiered approach. I do biomarkers and clinical trials, and in my world, the context of use for a biomarker assay will change. So we want you to have your initial validation appropriate to that context of use, but we want it to be good enough so if your context of use changes, you can build on that. You don't need to start over. So that's that in a nutshell. Oversimplified, but there's two types of validation: analytical method validation and clinical validation. And in the method validation, we establish which measurements the method measures. We'll have a drinking game tonight and see if we can all say that. But basically, what cells are we going to report, or what are we reporting in this assay? And then we look at what is the level of performance achieved. You just saw some really nice data from Jolene, and she's got like 3% CVs in her assay. We also, in a validation, look at what are the limitations of our assay. I know all of you doing leukemia lymphomas know, okay, if we have a patient treated with anti-CD38, we can no longer use that in our assay. Only when you have an analytical method validation can you do a clinical validation. And that means, what's the benefit of the test result to diagnosis, prognosis, or clinical outcome? And I think Oral will be addressing a little bit of that, I imagine, in his presentation. So now I'm going to share with you 3 case studies. The first will be how we understand validation terminology and how that translates in flow cytometry. So accuracy is a big part in these two papers, which came out the first in 2011 from the American Association of Pharmaceutical Scientists, all of us biomarker people. And the second, I was involved with both, but from International Society for Clinical Cytometry validation guidelines. And we talk in these papers about the challenges of doing accuracy in the traditional sense with flow cytometric methods. Now I'm going to show you this paper here, and I think it's a very good paper. These are actually friends of mine, and they had an assay, and they are using this assay as an enrollment criteria in a clinical trial, because that's the context of use. They've got a greater validation burden than we would for an exploratory biomarker. The assay is pretty simple. We have, I think it's a kit from BD actually. They're gating forward and side on monocytes and then cleaning that up with CD14. But their reportable result is the level of expression of HLA-DR on the peripheral blood monocytes, and they're quantitating that with the BD QuantiBrite beads. So again, the context of use is an enrollment criteria. For QC, they've done what we have to do in flow cytometry validations, is they had to get creative. They're using the STREC QC, which does have target values for the CD14 monocytes, but this is their marker. So what they've done is exactly what you should do. This is really similar to the matrix of their assay, whole blood. It's sort of a contrived whole blood control. And they established their QC ranges. They're using the QC to monitor their assay. Now, the laboratory, the testing laboratory is in New York State. And so they need to have approval because it's an enrollment criteria from New York State. I just downloaded this the other day. So their guidance document for validation is 2011. Now, lots has happened since then, and these validation guidelines have not been updated. So the commercial controls, use them when possible to demonstrate accuracy. When available, use the same matrix, and your results must be consistent with the manufacturer's assay values. Now, this QC material does have target values for the CD14 monocytes, but that is not what is reported in this assay. It's an upstream gate. So in order to check that box to fulfill whatever, New York State had them demonstrate accuracy to a value that they are not reporting. It is a midstream gate in this gating strategy. And I just question this. What is the value of that? How does that tell you the accuracy of your assay? And the answer is it does, of course. So we have the reviewers, no one understanding the assay, the validation. And so they're doing something that's pretty meaningless. So case study number 2 is how, again, the validation terminology translates to flow cytometry and understanding the context of use. So we'll go this, just a short talk, so we're not going into too much on the validation, but specificity, now called selectivity. I don't make up the terms, I just follow them. And so basically in flow cytometry, that means, are the cells in my gate what I think they are? And we don't really validate that. This occurs during assay development. And we heard a lot about this yesterday. Are we using the right phenotype for our population, our gating strategy, compensation, blocking reagents, blah, blah, blah. So we develop a really specific assay to measure the cell type that we want. And then we have sensitivity, which you don't always need to validate, but if you're, again, Jolene mentioned some rare events, then you need to know at what point my assay is no longer reliable. You no longer have precision. And this is influenced by your panel design a lot, like how you how many events you collect, but you can actually demonstrate that in validation. I won't go into that today, but we have a nice paper on that. So I found this paper, and they're using flow cytometry to distinguish different stages of tuberculosis infection. So in my opinion, this is the context of use is maybe patient staging, prognosis, care and treatment. So this should be validated for a clinical setting. So you need the analytical validation and the clinical validation. This paper had 77 references, some of them referring to the phenotype they use. So they're addressing specificity, looking at these markers and all, but not one paper reference in the document addressed validation. Their results, I believe, they're taking CD4 cells, activating ex vivo with mycobacterium, and looking at gamma interferon response. Perfect. You know, I don't have a question. This is adequate staining. Now here in their uninfected patient, they're claiming 0.53% positive, but they have not validated this method. They haven't evaluated what's my limit of detection, what's the noise in my assay, and when I have this value, is that true? Is that above the noise? What's the precision there? And what's the difference? What are the precision of all of these assays? So, or all of these reportable results. Yet they took these results and they gave them to the statistician, and they're claiming the significance when we don't have any validation data here. And I really question that. And so in this case, I think the laboratory itself doesn't understand the validation process, and they're making claims on their data, which I am not convinced they can make. The third case study is understanding technological advances and understanding the technology. This is our garden variety TBNK assay, which everybody uses. Jolene talked about in laboratories, and it's a Lysno wash assay, 6 colors, a single tube. There's automated gating, but you have the option for manual gating. And this is CE marked and IVD approved. So we look first at side versus CD45 gate on our lymphs. Then we're going to look at T cells versus non-T cells, CD4s versus CD8 in the Ts, and NK, CD16 and 56 in the same fluorophore versus 19. So you can get B and NK out of there. A little bit of history of this assay. The first iteration of it that I'm aware of is for the caliber, and it was a 4-color instrument. So you had to use 2 tubes, and you had to put CD19 and 56 in the same color because you only have 4 detectors, which was a cute trick at the time. And the second iteration was validated for the Canto, and later the Lyric, as Jolene shared. Now we've we're able to use 1 tube instead of 2, which is an advancement. That's good. But we still kept CD56 and 16 in the same color, which we didn't need to do. Now, in order to maintain IVD approval or obtain IVD approval on this new assay, for accuracy, the agency requires that you match data from a predicate device. So we've got a new instrument and arguably more sensitive, but we're making sure the assay fits this older technology because we want to get IVD approval. So it's just, I think they miss an opportunity to give us a better assay. I'm not saying this is hurting patient care at all, but this assay is not as good as it could be, but it's IVD approved. They could have, I'll show you in the next slide, added CD14, maybe even CD235. They could have had the opportunity to report additional results, such as 56 on T cells, different NK cell subsets, but they kept these fluorophores in the same color. These data are from my friend Stacy League at Mayo Clinic, and just illustrates exactly what I was just saying. In this sample, it's not a good quality sample. The auto gating's off, and it's including CD14 in the lymphocyte gate. So that means all the other gating is very much off, and you can see here in the the CD4 gate, we're looking at monocytes and not just T cells. So she had a poster one year at ICCS showing if you add CD14 and just use your own gating strategy, you can have a much more specific assay. Now, this breaks the IVD clearance on this, and now it's become a laboratory developed test, but arguably a better assay. And at one place I worked, the medical director was not a flow person and was really skeptical every time we did not use the auto gating. And when I came up with data like this, I would show him and say, we have to use the manual gating. So it's just this misconception that IVD means it's the best assay because it's cleared by the agency. Okay, now just a few random rants. The document CLSI H62, Marcia mentioned the Document Development Committee. We're 37 members, 37 experts in the field. And under CLSI, there's lots of rules you follow, which is why we like having the weight of CLSI behind the guidance documents. And one of the things is your composition of your committee. You need members from the government, industry, and professionals. And so from the government, we had an FDA rep and a rep from NIST. The other thing is the document goes for public comment, and you need to address every single comment. The people submitting the comments can appeal, and you need to address the appeals. And all of that back and forth is documented. It's publicly available. The FDA was part of the writing committee. We got lots and lots of comments. They had 10, I believe, and we went back and forth with them, and finally they accepted our resolutions. But they still have not, in their language, recognized this document. And we say the FDA, but there's actually many, many different branches and offices of the FDA, and like any big organization, they're not necessarily talking to each other. From my perspective, the parts of the FDA that are important to this document are CDRH, and they're the ones that would really regulate devices. It's so cute, they call assays devices, but they oversee that. And then there's the Center for Biologics Evaluation and Research and Center for Drug Evaluation and Research, which also can evaluate biologics. Because I'm doing biomarkers supporting drug development, all of these agencies are important to me. So I'll have to tell you that people from CBER as well as CEDAR have said, oh, thank you for working on the document. We use it all the time. And I'm like, well, why hasn't the agency recognized it? And they're like, that's beyond their pay grade. That's not part of their job description. They have no idea. But they said it's useful and they're using it, which is great. But when we go back to what we're going to talk on our panel discussion, if the agency wants to have oversight, we need guidance, right? And we have a very good guidance document out there. And still not officially recognized. And the lack of recognition is hurting the field. It makes it harder for laboratories to implement what we've put in the document. Validation is not cheap. Doing QA is not cheap either. Flow cytometry is expensive. And laboratories have told me that their QA department are not flow people. They don't recognize H62 because the agency has not recognized it. Other people have said, you know, if the agency recognized the document, I could get the resources and funds I need to do these validations. So they're hurting the field, and we addressed all of their comments. Getting back to New York State, again, what we're talking about, the reviewers need to be current in the field. And yeah, there's still, this was from 2019. They're telling you how to validate an assay. And they're still recommending isotype match controls. And here for CD45 versus side scatter, that's going to, you don't even need an FMO for that. It's going to give you no value. And since I think 25 years for gating controls, FMOs have been recommended and not isotypes. So closing remarks, both the testing labs and the reviewers need to understand flow cytometry validation, and they need to understand the current best practices. And this slide, I was told I was never to give a talk and not show it. And my friend Ira from MetroFlow, which was my first committee ever, created that for me. So thank you. I think we're gonna have the. Oh, are there any questions that we also have the panel later? I think.
So I know we're gonna ask some questions during the roundtable, but Virginia, just real quick, I know you know the FDA, or you acknowledged in your talk that the FDA hasn't yet recognized that document officially. Is there anything else that could have been done for that? Like, what do you think the missing step is in getting anything recognized so far? I mean, because we've been inclusive, right? They've been part of the conversation.
No, and they're really part of CLSI. So I'm on the governance of CLSI. There's expert panels that oversee these documents. I actually have a call at noon for the expert panel for this. So the expert panels, the governance, there's FDA reps throughout. And the comments we got from the agency, there were some nomenclature, and it's like, okay, in pharma, we call it this. FDA likes that. And I did learn one thing, that on nomenclature, they are very careful to use the same terms that are in the laws. Oh, and it's like, okay, that makes sense. So we made those changes, and actually we employed the fit for purpose validation approach, so that even if you weren't submitting for an IVD cleared assay, you could have a reasonable validation. And they actually, I was surprised, they were okay with that. So their comments were really around nomenclature. And then it got funny, because I was like, oh, I don't care what we call it. So, you know, changing nomenclature. And then NIST was like, no, NIST doesn't recognize that term. But on the fundamentals of what we were asking. And we did put in some language there, because they were nervous about if we're recommending number of reps, number of samples, which I never see in validation documents. And I really wanted that in here, because otherwise it's useless. And I see so many validations that people do that are a little bit misguided. I want more testing on this and less there. Or this with the accuracy. I've seen so many crazy things with accuracy. So the other thing the agency said was, you know, we put a lot of disclaimers in, and I cut and paste this term all over the document. You know, these are minimal, reasonable requirements for validation. If you're going to submit to an agency, you know, talk to them.
Yep.
And then one person was like, oh, well, then the document, you don't need the document at all. And I was like, we do.
Yeah.
So I don't know. I don't know.
Yeah, one other quick question, right? So as we move into personalized medicine and medicine becomes more unique to the patient, we had a webinar a couple months ago where we talked about how do we actually, you know, try to line up what's happening in clinical research and roll it over more easily into the Jolenes, right? Where you can kind of actually upstream and downstream have some connection points. So are there other documents that are needed for that? I mean, is that like a next step, or how do we actually tackle? And that's, I think, always, right, decade long, great question. How do you bridge translational? But in personalized medicine, it becomes, I think, even more necessary to keep that gap as small as possible. Yeah. Okay. Let you guys break for a few minutes, go grab some coffee, and we'll be back for round 2 of the clinical session. Thanks for your attention. All right, welcome back. We're going to go ahead and get started. I know people will be filtering in, but just for the sake of time, we're going to get started. My pleasure now to introduce you guys Dr. Alpan. He comes to us all the way from Virginia, right outside of Washington, D.C. I met Dr. Alpan, gosh, probably, I don't know, 7, 8 years ago at an AAI conference. We were looking at allergy. As we know, flow cytometry has been very crucial in oncology, but it's actually expanding into allergy and autoimmunity. And the one thing that I want to tell you that's not on this piece of paper where you can read about Dr. Alpan's career is that my daughter actually, when she was about 13 years old, started to struggle with signs and symptoms. And we went for years without a diagnosis, but then very quickly it became problematic where she would hemorrhage blood and we would be on long doses of steroids. And Dr. Alpan really, many of my friends stepped in and said, you know, how can we help? But Dr. Alpan really went the extra mile and not only ran some flow cytometry himself, but sent me to his good colleague at Cincinnati Children's. And that extra information changed her prognosis. We were able to get on the right medicine at the right time, which is actually really crucial. She did end up with a Crohn's diagnosis, but had it not had all of the information along, I don't think that they would have been able. She was actually the first pediatric patient to go on a drug called Stelara, and it changed overall the way that Stanford treats pediatric patients. And so for me, there's a huge testament in my life that flow cytometry is essential, but beyond that, that looking holistically at a patient and bringing in all the different variables can actually help you narrow down what is, and in her case, what isn't happening. And so just really thank you for what you did then, but I know Skylin is only one of many patients. Dr. Alpan has also received private funding from senators to go after additional testing. And so he's really a patient advocate first and foremost, and really excited to hear what you have to say today.
Thank you very much, Marcia. Thanks for the invitation. In October of 2023, late at night, as I was drafting my response to the proposed FDA ruling on regulating laboratory developed tests as devices, an interview with Alan Dershowitz unexpectedly appeared on my TV screen. Hearing Alan Dershowitz talk on constitutional matters, as he usually does, prompted a realization that the issues at hand extended beyond just regulatory concerns, but to encompass broader constitutional violations by the FDA as well as safety issues. Motivated by this insight, I revised my response to the FDA's proposed ruling. Fast forward several months, on a Saturday morning, I was enjoying my coffee, watching the view of the Chesapeake Bay, where it was a historic gateway for colonists to the United States. I decided to browse the FDA comments on the word Constitution to see if anybody else has written about that. I discovered Dr. Barbara Evans's work, who will be giving a talk shortly here also, and her comments to the FDA. I reached out to her the same day by email, and to my pleasant surprise, she responded immediately, expressing her attempts to contact me. And over the past few months, we've had many discussions, and now we're here together presenting our collaborative findings. So I've never anticipated delivering a speech like this, one that defends the very practice I was trained for. It's straightforward in that I'll share you my personal journey throughout medicine, but yet it's challenging because if the proposed FDA ruling is adopted, it's as if my role never existed. The proposed rule seeks to amend the FDA's regulations to make explicit that tests, laboratory developed tests, are devices under the Food, Drug, and Cosmetics Act. My talk isn't an exhaustive overview of the FDA regulation, but rather it's focused discourse on an overlooked area of medicine, one that's neglected by the FDA, most lawmakers, and most government officials. Congressman Dr. Burgess of Texas highlighted at last week's House Energy and Commerce Committee hearing that we physicians don't rush patients into surgery to remove an organ based on a single test, FDA approved or otherwise. The debate on laboratory-developed tests has lost its direction, resulting in potential ethical, constitutional, as well as patient safety issues. The proposed regulation also fails to value human life, the lives that may be impacted or lost if this rule is finalized as is. This morning, I will detail the medical concerns, and my colleague, Dr. Barbara Evans from the University of Florida, will address some of the constitutional aspects. While I will concentrate on my area of expertise, which is immunology, flow cytometry, the principles apply to countless specialists and millions of patients across the country. How do I move? Okay. So these are my disclosures. Right. So let's start with some basics here. So when a physician sees a patient, their decision-making process typically begins with gathering information through clinical history and physical examination. They listen to the patient's concerns, symptoms, and their medical history. They observe vital signs, and they observe the patient's physical condition. So based on this information, the physician formulates a differential diagnosis, which is a list of potential diagnoses that could explain the patient's symptoms. Then they prioritize this list based on the likelihood of each condition. Diagnostic tests, such as blood work, imaging, or other procedures, may be ordered to further narrow the diagnosis. Through this process, the physician considers the patient's overall health, risk factors, and also preferences. Once a diagnosis is made, the physician develops a treatment plan, which might include medications, therapy, lifestyle changes, or referral to other specialists. This plan is communicated to the patient, ensuring that they understand the diagnosis, the treatment options, and expected outcomes, allowing for an informed consent and shared decision-making with the physician. At no part, us physicians act on a single test results. The discussion on LDTs have shifted to this artificial assumption. Diagnostic tests, they're crucial components of decision-making in healthcare. But, there's a big but, they require interpretation in the context of other tests, whether they're laboratory or not, and clinical data. They're not used alone in diagnosing patients. So I'll give you an example from my own specialty, allergy immunology, that shows diagnostic tests are not infallible. So consider this 51-year-old attorney with a history of severe anaphylactic reactions to all forms of shellfish. And it's to the extent that even the smell puts her into anaphylaxis that she has to use epinephrine. If you look at these test results, you see a number of shellfish on here. They're pretty negative. However, when you look at her, she suffers reaction. So who's wrong here? Is the patient wrong? Is the test wrong? The manufacturer wrong? Is the FDA wrong? And the answer is none of them. Nobody's wrong. Biology is not black and white. So these tests are merely a component of a comprehensive diagnostic process patients go through. So if biology in medicine was black and white, there would be no need for medical professions, and instead a visit to the local hardware store would suffice patients' healthcare needs. In the diagnostic process, the patient is always right. As physicians, we always ask the question, why is this person sick? What's behind the pathophysiology? That's why we go through testing. But the problem is, if the patient dies, and even if you have the best explanation to negate that, the patient is always right and remains right. So we have to be very careful here. I put this algorithm up here. It starts with the patient and the provider, the interventions we do to the patient to understand what might be going on, Then comes tests, test results, and then everything else, regulations, hospitals, meetings, etc. It's only when things are done in this order, it becomes safe for patients. And the FDA's proposed ruling is turning this flowchart upside down. It starts with regulations, everything else, and then tests, and then it goes backwards. This is starting from the wrong end of treating and approaching patients. It's wrong and it will be dangerous. I want to briefly go over with you some organizations so we kind of have an understanding for those of you that are not very familiar. There's a Food and Drug Administration. Their role is they ensure the safety and efficacy of medical devices, including in vitro diagnostic products. And their mission is to protect public health by ensuring safety and efficacy. Then there's CLIA. This organization establishes quality standards for all laboratory testing to ensure a lot of the different technical aspects of the testing. And then there's CAP. It provides laboratory accreditation based on very rigorous standards, and their goal is to aim, their aim is to improve patient care. And they foster excellence in the practice of pathology and laboratory medicine. But one thing that's interesting about the FDA's role is I talked about efficacy and safety, which is very, very important, but it serves a very narrow need. So meaning when we're trying to get a drug approved or a test approved, anything approved, we have to have a disease. We have to have inclusion-exclusion criteria. So we have to pick certain patient populations in that disease. And we have to have an endpoint, not multiple endpoints, an endpoint. But the problem is we have, in my specialty, hundreds of diseases. And when you look at across different medical specialties, there's thousands of diseases, and there's diseases we don't even understand that well. So suddenly trying to use this process that the FDA has to apply to every single disease, every single condition, every single patient symptom, it's just not going to happen. And as physicians, when we make decisions using the tests that we use right now with laboratory developed testing, we use our experiences, we use published guidelines, we use things that we learn in conferences, our communication with our colleagues. There's a lot of things that happen that allows for the decision making. But there's two other organizations we forget about when it comes to regulation. One of them is Health and Human Services, and the other one is medical boards. So if you look at the mission of HHS, it's very different than the FDA, even though the FDA is an organization under HHS. The role of HHS is to enhance the health and well-being of Americans, just like us physicians. We try to make our patients feel better, and get back into society, their lives, and their professional work. And then there's the medical boards. They regulate the practice of medicine within a specific state or jurisdiction. And they regulate everything we do, not just what we give patients, how we treat patients, but our conduct during our patient encounters. So there's things beyond the FDA CLIA cap that there's regulations for both from the HHSs and the medical boards that the practice of medicine is regulated. And one might think about, how does HHS get into this whole thing? It might be a little confusing to you. CMS is under HHS Medicare. And when Medicare publishes guidelines, rules, and how medicine is paid for, a lot of the commercial payers follow those guidelines. So even if though the HHS through CMS may target a specific elderly patient population, their input becomes nationwide and across all ages at the end. So this slide is very important, but let me explain this to you a little bit. In discussing the testing process, we consider three distinct phases. The initial phase is the decision to conduct a test. So this is the part where we decide what test do we order on the patient. It's made by the healthcare provider overseeing the patient's care in consultation with the patient. So this stage, as you see on the slide, is called the pre-analytical phase. Following this comes the analytical phase, which we talked about at the previous two talks, all the validations, technical aspects, machines, etc., which is very well regulated at the present time. This stage is under the purview of CLIA and various private entities like CAP, COLA, etc. The final phase involves decision-making, where the provider integrates all the information, the test results, the clinical findings, to come up with an informed decision about the patient care. The results may come in the form of a consultative note, like one from a pathologist, or simply as raw data certified by the pathologist. All tests, they be laboratory-based, radiological, or other, are akin to tools in a physician's toolkit, aiding in the determination of the appropriate course of action. The testing process is complex. It involves multiple components, some of which are manual. To attempt to streamline this process into a one-size-fits-all solution will be futile, akin to forcing a square peg into a round hole. Dr. Evans will dive a little bit deeper into this process after my talk, but she will most importantly offer solutions that will not collapse the medical care for patients and the practices for physicians. Laboratories can be in different places. It could be independent laboratories. It could be in hospitals. It could be in doctors' offices. They're called in-office labs. There's a lot around the country. One thing that has been interesting is in the FDA's proposed ruling, there's no mention of in-office labs, as if they don't exist. Now, I'm going to switch gears a little bit to put things into a little bit more context for you on how does a medical trainee integrate into the current healthcare system in the United States. And I'm going to illustrate that by using my own journey. I completed my pediatric residency at SUNY Stony Brook, where I developed a keen interest in biology of blood cells. My research during the residency led to an invitation to present at the NIH Research Festival. There I had the opportunity to engage with many scientists from the NIH intramural program and was deeply inspired. Michael Gottesman, at the time, who was the intramural director of the NIH, encouraged the 25 residents that attended that research festival to pursue a path combining science and medicine at the NIH. So I followed his advice, and about a year and a half later, became a clinical allergy fellow at the National Institute of Allergy and Infectious Diseases. This was an extraordinary environment that expanded my horizons in medicine. I learned the importance of research in conjunction with clinical care. At NIAID, I witnessed firsthand how assays are developed and that could lead to discovery of new diseases and new treatments. Post-training, I was encouraged to extend this knowledge and practice. I continued at NIAID for several additional years, working under the mentorship of the esteemed scientist, Polly Matzinger, whose teaching profoundly influenced my thought process. With that foundation, I was prepared to venture into the broader world. Now, one of the things that I first saw when I was in clinical practice is how much we don't know. This is an iceberg. You know, we always think that patients walk into an office, they fit into a textbook, we make the diagnosis, they take their pills and they leave. That does not happen. There's a lot more we don't know than what we know. And patients come to our offices from different pathways. Sometimes they come with symptoms, with clinical care that they have had elsewhere. Or they could come to our offices with an abnormal diagnostic test result, whether it's a blood test, X-ray test, whatever it might be. Or they come through education and advocacy. It might be that we may be giving a talk at a local medical society meeting, and it might spark the interest of an internist, a pediatrician, or another specialist thinking about immune disorders. So patients come to us from very, very different directions. When I first started in Northern Virginia, there were problems. There were large hospital systems, no diagnostic immunology lab to support the diagnosis of inpatients or outpatients. This had led to a backlog in Northern Virginia, and we're talking about a place that's 10, 15 miles away from Washington, D.C., a very urban area with a lot of resources, one would imagine. But also there was unwillingness of the local health systems to refer tests out of the state. So I was stuck. So what was the solution? I set up an immunology flow lab in Northern Virginia over a decade ago with personal resources and with some of the most experienced staff in the country, which I dragged from the National Institutes of Health. I got it capped and CLIA approved in 2011, set up immune symposia, educational session, did local networking in the community for educating the community physicians for immune evaluation. But most importantly, the laboratory became the heart of my medical practice. The way I was trained to think was now influencing what I do. And there's thousands of practitioners around the country that has gone through the same process and is still going through the same process that I explained to you. So the triangle I showed you with the resources that we created through laboratories, it grew. It grew into research and translation, education and training of residents, other physicians, community outreach, not just for other physicians, but patients, as well as diagnostics in clinical care. So now you have something really plugged into the local healthcare in where I am. And this happens all across the United States. So what does that do? It allows access. It allows access for physicians, allows access for patients. It becomes very cost effective to do. It's very fast. The speed is there. So the patients don't wait and wait and wait and wait to get answers. It builds a patient-physician relationship. It builds also relationships between physicians in a given community. But also, more importantly, by diagnosing patients a lot more rapidly, it brings safety to healthcare. So then came along the proposed FDA regulations. So these regulations carry implications for safety. They violate constitutional rights, and they place a value on human life that they fail to adequately address. The proposed regulations state that small organizations like mine will go away. They explicitly state that in the proposed regulations. This is not only wrong and unconstitutional, but also very dangerous. Moreover, the repercussions will also extend beyond to healthcare providers. They disproportionately will impact certain patient groups, especially those with rare conditions or targeting specific ethnic demographics because of the pattern that some of these rare diseases are seen. And I published a small letter in the Washington Post. This was in October, about the discriminative effect of such a proposed ruling. Now, before going into the next presentation by Dr. Barbara Evans, I asked 3 of my patients. 2 of them were mothers of 2 patients that I had, and one of them is the patient, to give their own perspective to you in 1-minute video message on what they think about this all. So you heard from me. Now you're going to hear from 3 patients. Let's see if I could get this to work.
Hi, my name is Betty Walter, and my son John in 2009 had a bone marrow transplant. We spent about 12 years trying to get him properly diagnosed as he continued to get sicker and sicker. And it was the ability of a research doctor with a lab that did flow cytometry analyses that was critical in helping to diagnose John. The reason that parents like us brought our sons to that kind of doctor was because as a researcher, he had the ability to create laboratory tests unique to the type of illnesses that John's fell into. And I have no doubt that without those tests as developed by that doctor, John would not have been properly diagnosed and would not have gotten proper treatment and probably would have died. It was a very critical situation. And I knew many other families in that process who benefited because of the ability of doctors to innovate in the lab. And I have intense concerns about the FDA coming in and telling people like my family that a one-size-fits-all lab like Quest Diagnostics is the way to go. We were not well served because of the unique situation that we faced. And I think any family that is trying to get a solution is thankful for the freedom that researchers and independent labs have to cater to the specific needs of their patients. Thank you so much for considering my thoughts.
Hello, my name is Colin Seal. I am a 39-year-old common variable immune deficiency patient. I am married and have a about to turn 5-year-old son who, thank goodness, as of right now, seems to be pretty healthy. But without the role that the doctors and the innovation and the thinking outside the box of the group of medical professionals I've been able to see over the years, I don't know that I would be able to say all those things I just said. I've been able to have a successful career. I've been able to marry the woman of my dreams and have quite a wonderful life. But that wasn't the case years ago. You know, it took 14 years for me to get diagnosed. Commonly was going to small doctors who were following out-of-date guidelines, who said, if the person presents in this box, then this is how you should treat them. The problem is the way medicine is developing today, and for patients like me, we don't always fit inside a box. You may hear me breathing rather harshly, and that's because of years of doctors trying to tell me I fit inside this one box and putting me on multiple doses of steroids like they were told to do for patients that they put in that box. I now have scar tissue that is built up on my right lung. Now, it's important that we continue to allow doctors to do what they think is right and to innovate. You know, a lot of times people say, well, that's why we go to these big medical centers. And I was seen by one of those medical centers here in Baltimore, Maryland, where I live. And for years of being seen at this large medical center, I had about 3 weeks where I was bedridden. I had fever, cough, could not work, was not able to do my job. And this went on for 3 weeks. And every time I called my immunologist, I was told it's viral. It's viral. And yet again, one of the largest institutions in the country who's world renowned was telling me you fit inside this box and here's how we're going to treat it. Ultimately, I had to make a decision. I made a doctor's appointment with this leading institution. And again, they said, hey, we're not going to waste medical dollars in the system trying to figure out, you know, just to see that you have a viral issue going on. So here's a patient who's 3 weeks bedridden and is being told that, you know, again, you fit inside this box. And finally was able to find a local physician I had worked with previously in my career. They were able to take me on as a new patient, and through some innovation through a private lab, we were able to discover the reason I was having so many problems is because I was having an inflammatory attack. And really what we needed to do was start, you know, kind of a unique dose of medicine. And after finding that out, we've been able to treat it. Through innovative labs, we've also been able to discover that, you know, I'm a rather unique common variable immune deficiency patient inside the dose that I take. If I took the dose of IG that the box says I'm supposed to take, I don't know that I'm alive today. Without the innovation of my medical team, without them being able to think outside the box and not just label me as what they're told to do through regulatory bodies, I don't know that I'm here today. And if that were to change and the FDA were to get more involved, I'm scared that my son may not have a father because we're stripping innovation out of the people who help keep us alive. Thank you, and look forward to continuing the conversation.
Hi, my name is Nicole Carver, and I'm here today to share with you that with the use of extensive laboratory-developed testing, my son Hudson has been able to receive treatments that have helped him outlive any textbook prognosis. With these innovative diagnostics, Hudson went from a prognosis of having zero quality of life, being told he would never walk, talk, have a trach, and most likely not live past the age of 2, to turning 7 years old this year and paving a new path for many children to come. Along with fighting against a terminal and incurable genetic disease, Hudson has had to overcome countless admissions to the pediatric ICU for respiratory illnesses from 2018 to 2022. With the use of innovative laboratory testing, Hudson started receiving additional treatments June of 2022, strengthening his body's ability to fight off respiratory viruses and drastically reducing his hospital admissions to twice in the last 2 years related to respiratory illnesses. Without the ability to test beyond the perceived known outcomes, my child quite possibly would not be living the life he is living today.
Thank you.
So we are gonna have a roundtable after this where we go a little deeper into the topic, but just making sure there's no immediate questions. All right, so Sarah might need you. Are you gonna be able to share from there? Okay. All right, so while she comes up and does that, I'm going to introduce Dr. Evans. So we have yet here to have Dr. Evans speak for us, but I think that today's talk will not be the last from her. She is, as Dr. Alpan alluded to, she practices law. She's currently actually out in Florida at the University of Litwin College of Law, but she was actually trained in Texas. She holds a BS in electrical engineering from the University of Texas at Austin, an MS and a PhD from Stanford University, a JD from Yale Law School, an LLM in health law from the University of Houston Law Center, and she completed her postdoctoral fellowship in clinical ethics at MD Anderson Cancer Center. So she really wanted to be with us today, but she's actually at the White House. One of the things that she's currently working on is actually the legal studies team for the NIH Bridge to Artificial Intelligence, patient-focused course for equitable AI. It's a project that they're using to develop the national data infrastructure for AI in clinical care. So I look forward to welcoming her to Houston in another year, because I think she'll be able to even share with us in the future about AI. But for today, she's actually going to really bring in some history because she's going to be talking a bit about our rights and the First Amendment. So I'm just going to press play.
My name is Barbara Evans.
Real quick, can they hear online? We're sharing this sound? Okay.
I'm a professor at the University of Florida. I'm so sorry I couldn't be there in person today. I actually have my primary residence in Houston, about a couple of miles from where you are, so I had really hoped to attend, but I simply couldn't. I have no commercial conflicts to disclose, and the views I'll express are my own scholarly opinions. This is the First Amendment to the U.S. Constitution. Congress cannot make laws that abridge the freedom of speech, and Congress cannot authorize federal agencies like the FDA to do so. And this same thing applies to state governments through the 14th Amendment. How does this apply to a particular type of speech? What is professional speech? This is expert advice that a licensed professional gives while providing regulated professional services to a particular patient or client in the professional setting. The paradigm case of professional speech, it would be a physician providing expert diagnostic or treatment advice for a patient when the purpose is for clinical care. And obviously, this encompasses a MD laboratorian when preparing a test report for a particular patient. I've listed a couple of things that are not professional speech. If a doctor goes on the radio and says everybody should eat broccoli, that's not professional speech because it's not targeted at any particular patient. And in the same way, if a doctor meets a patient somewhere outside the clinical care setting and gives advice that's totally unrelated to healthcare, that would not be professional advice either. The question is, how does the Constitution address professional speech? These are some basic facts about the First Amendment. There is a whole amount of speech that the government can freely regulate, examples like fraudulent speech, child pornography. These are dangerous types of speech that the Constitution allows the government to regulate. But I want to point out the mere fact that speech is untrue or subject to high uncertainty doesn't automatically make it dangerous enough to avoid constitutional protection. If the government passed a law that said nobody can tell a lie, we would all spend so much time fact-checking that we would never be able to say things that might prove true or useful. The constitutional speech protection kicks in the lower part of this chart. And I'm going to start at the bottom. That's the most protected, fully protected speech. Things like statements of religious belief, scientific opinion, art, literature. This has the highest protection, which we call strict scrutiny, which means a court reviewing a regulation that restricts this speech would be very strict in protecting the speech. In the middle is something called commercial speech. This has been protected since the 1980s. It's things like advertising, medical product labeling. It receives an intermediate level of constitutional protection, which is still quite significant. For years, there was a debate about where does professional speech fall on this. Some people said it's regulable because medical practice boards can sanction a physician for giving bad advice. Other people said, no, this physician expert opinion is more like scientific speech and it should get more protection than that. This question finally got resolved in 2018 in this Supreme Court case, which held that the First Amendment does protect professional speech and it receives either intermediate or strict protection. The court said it wasn't deciding which it was, intermediate or strict, because in that case, the regulation wouldn't have been constitutional under either of the standards. So that issue has not been resolved yet. How high is the protection? But it gets a lot of protection. Now, there's some things I want to tell you from long-standing Supreme Court precedents. First, the First Amendment doesn't just protect speakers, it protects listeners and users of information. So it would protect against government interference with information flows that speakers need to tailor their own remarks to the needs of an audience. Things like having access to the output of a LDT to inform a test report that a doctor is preparing for a particular patient. That would fall under First Amendment protection. If the government restricts access to informational inputs that people need to develop and tailor their own remarks, that is content-based regulation of speech. For example, if the government told me I cannot read any law books when I give legal advice, that would alter what I can say and it would regulate my speech. And content-based regulations are generally unconstitutional, and the government has to bear the burden to prove why they're justified. Now, the way I look at this, LDTs supply informational inputs to the speech of laboratory personnel when they're preparing a final test report. The lab isn't making that test in order to sell it as a product. So it's not really subject to product regulation. It's being used internally to inform the professional advice of the laboratorian. And if the laboratorian is a licensed physician, it clearly seems to fit within professional speech. I'm not saying that this same protection doesn't extend to non-physician laboratorians. They may be protected. We just don't have case law on that I was able to find. The FDA's proposed regulation of LDTs would restrict laboratorians' access to information flows to inform their speech. It would require a period of time when you can't use a test until it has gone through pre-market review, and could delay access. The First Amendment actually requires the government to bear the burden of proving that its regulations are justified. So when you have the First Amendment implicated, the lab doesn't bear the burden to show that the LDT is safe. Rather, the government bears the burden to prove it's unsafe before the government can block or delay access to it. In our legal system, medical professionals, rather than federal agencies, are the ones who decide what is the standard of evidence that is appropriate for a doctor to consider when tailoring professional speech to a particular patient. That's long established in state and federal law. Now, when speech is protected by the First Amendment, that doesn't mean that it can't be regulated at all. It means simply the government must design its regulatory scheme very carefully and within certain constraints. I'd like to show you what the constraint is for that intermediate level of protection, the commercial speech protection. This is the so-called Central Hudson test, which is named after a case where the state of New York was restricting advertising by Central Hudson Electric and Gas Company. The test is that the government can freely regulate speech if the speech is misleading or concerns an unlawful activity, such as encouraging people to sell illegal drugs or something like that. But the mere fact that something is scientifically uncertain or debated does not automatically mean it's misleading. That's been well established in a long line of FDA cases, that just because something's uncertain, that doesn't mean it would always be misleading to people. If one is not the case, the government can't freely regulate speech. It can regulate it, but the regulations have to meet certain conditions. The one is that the government has to prove it has a substantial interest in regulating the speech. And the government needs to come forth with data showing this speech is harmful and we need to regulate it. The regulation has to directly advance the government's interests. It needs to be directly related to the problem that the government is trying to solve. And the regulation can't be more extensive than necessary, which means it can't restrict more speech than it has to get the job done. So that means things like don't regulate and ban speech altogether if it would suffice just to give consumers a warning to tell them this is uncertain or you should consult, get a second opinion. You don't ban speech when warnings would work. And importantly, you don't ban all speech to prevent a subset of speech that's provably harmful. I think we all know that there are some lab-developed tests out there that are careless and dangerous. That fact does not entitle the government to limit all lab-developed tests, including those that can be extremely useful and important to have. You can't have an overbroad regulation that regulates everybody to take care of a few bad actors. Interestingly, the CLIA scheme has struck a balance that strikes me as being satisfactory under the First Amendment. First, CLIA regulates analytic validity, but not clinical validity or utility. That's important because there's a long legal tradition, and I think everybody agrees that an analytically invalid test is dangerous. So the government clearly has a substantial interest in regulating communication of analytically invalid tests. Importantly, CLIA avoids placing a prior restraint on speech. A prior restraint is when you tell someone you can't speak at all until you do this, this, and this. Non-prior restraint would be to say, you can go ahead and speak, but after you've spoken, if what you said is dangerous, you might get sued or you might face some sanction for it. But you haven't limited the speech with a prior constraint. And CLIA works that way. A lab can start using an LDT, and the analytical validation is reviewed by the regulator as part of the routine biennial survey after the laboratory is already allowed to start testing. This avoids suppressing speech before the government has clear evidence showing that the speech is analytically invalid. So it satisfies that constitutional burden that the government has to show why it's regulating. CLIA struck a balance that satisfies the First Amendment in another way. It avoids regulating clinical validity. Statements about clinical validity are scientific and medical expert opinion and enjoy a significant degree of constitutional protection. We don't have a long established legal tradition of letting the federal government tell doctors what their opinion can be and what scientific data they can consider. CLIA doesn't try to do that. It doesn't regulate clinical validity. Another point is that CLIA offers an option between direct governmental standards or a private alternative, an industry or professional self-regulation alternative. To offer LDTs, labs have to meet the CLIA requirements for high complexity testing, but there are two ways you can meet that. You can choose to have a certificate of compliance, in which case you're voluntarily agreeing to let the government set standards, or you can opt for a certificate of accreditation, in which case you're saying, no, I'll follow standards set by professional bodies such as CAP. The fact that you're not mandatorily subjected to governmental regulation is really important because it means you're not being forced to let the government regulate your speech. To conclude, I believe that FDA's proposal to regulate LDTs raised some serious constitutional concerns. Even if Congress granted FDA some additional statutory authority, that wouldn't solve the problem because the problem's constitutional. Neither FDA nor Congress can make laws that would violate the First Amendment. CLIA seems to offer a constitutionally sound approach for regulating LDTs, but as we all know, CLIA doesn't work perfectly. The Government Accountability Office has found great weakness in how it's enforced, and there are a lot of practical constraints, such as lack of proficiency testing material for some modern and novel types of tests. Nevertheless, strengthening CLIA might be a better approach to solving the problem of bad LDTs rather than using FDA's proposal to let FDA regulate MD laboratorians' professional speech. I'm so sorry I couldn't be there in person and wish you a really productive and interesting conference. Thank you.
So certainly eye-opening. Sarah will come up and help me. So next joining remotely is Dr. Jonathan Genzen, and he's actually coming to us from ARUP. You guys probably remember Dr. Ng's talk last year from ARUP. So they're a lab that continues to exude excellence, both under Dr. Lin and Dr. David Ng. But Dr. Genzen comes to us because of his role at ARUP currently. So he previously was a COO, their Chief Operating Officer, but has since been promoted to their Chief Medical Officer, and he's their Senior Director of Governmental Affairs. He is a professor as well at the University of Utah in the Department of Pathology, and he completed his undergrad education at Northwestern, and both his PhD and MD came from the University of Chicago. So with that, I will turn it over to you.
Thank you very much. Can you confirm that you can hear me okay?
We can hear you well.
Excellent. I'm going to share my slides. And if you can let me know if you can see the slides, I'll get started.
Looks good.
Did you say they're good?
Yeah, sorry, it looks great.
All right, thank you very much. It is an absolute pleasure to be here today. I'm sorry I'm not there in person, but I want to talk a little bit about our perspective on laboratory developed tests and the proposed rule and potential impact on clinical laboratories. As my disclosures, I am a professor at the University of Utah, and I'm Chief Medical Officer at ARUP Laboratories and Senior Director of Government Affairs at ARUP as well. Many of you may not be familiar with ARUP Laboratories, so I just wanted to pause here at the beginning to give you a little bit of a context on what our space is related to laboratory developed tests and clinical testing and what we do. We are the largest nonprofit clinical reference laboratory in the US, and we're a nonprofit clinical laboratory enterprise of the University of Utah's Department of Pathology. So we are part of the broader University of Utah system and family, but we're incorporated as a separate nonprofit, if that makes sense. We have hospital customers across all 50 states. We have over 4,300 employees. We're the largest life science employer here in Utah. And we do a lot of testing every day. It's over about 70,000 specimens on a busy day. And those are esoteric tests, not necessarily routine tests like CBCs or BNPs. These are things that people generally can't do in-house, and so they send it to a reference lab. We have an R&D institute with many R&D scientists and medical directors, and because we're part of a university of system, we are expected to publish and share what we learn. And so we've got a lot of publications, and many of them relate to LDTs. We have over 3,000 tests on our menu, and we've said this in our public comment letters, over 1,000 of those are laboratory developed tests. And so the LDT regulatory issue is a big deal for us and therefore all of our customers as well. So what did the FDA actually propose last fall? Well, it's 26,000 words in their proposed rule, but it's only a small sentence or two of what they're actually proposing to do. The FDA is proposing to amend by updating the definition of this term in vitro diagnostic product to make it explicit that in vitro diagnostics are devices under the Federal Food, Drug, and Cosmetic Act, including when the manufacturer of that IVD is a laboratory. So it's interesting. They're not creating a new definition of laboratory developed tests. Ironically, there actually is no federal definition of laboratory developed tests. Keep that in the back of your mind as we're talking. But rather, they're trying to imply that LDTs are IVDs if they're manufactured in clinical laboratories. And this is an important component here too. They believe that clinical laboratories are acting as manufacturers. Whenever I give a talk like this in person, I ask people to raise their hands if they think they're a manufacturing facility, and nobody in clinical laboratories ever does. So I think there's a discordance between what the FDA believes and what our own community believes. Along with the proposed rule, the FDA included a timeline of when it will roll out. I'm not going to talk about that timeline here, but everything wouldn't kick in immediately. It would be in several phases. So what is this law that they're planning to create this rule to essentially amend? The law is called the Medical Device Amendments of 1976. And this was the law that amended the Food, Drug, and Cosmetic Act to provide for the safety and effectiveness of medical devices. The law authorized the FDA to regulate in vitro diagnostic devices, and it established risk classes on how it would categorize tests. These are the class 1, 2, and 3 devices that we've probably all heard about. They roughly correspond to low, moderate, and high risk tests, but they don't really have a good definition of risk, which is something kind of interesting when you think about IVDs. The class 2 are the pre-market notification 510K pathway. This is the FDA clearance. And then the higher risk class 3 are the pre-market approvals or PMAs. And so you may hear the term FDA clearance or approval. Well, that correlates to class 2 versus class 3. And class 3 is much more expensive and burdensome. So we submitted a public comment letter from ARUP Laboratories that solicited feedback from a lot of our staff and employees here. We've also submitted letters to the Energy and Commerce hearing that occurred last week. And basically, anytime we can share our perspective, we're happy to. And so I'm going to share some of those concerns here as well. I want to emphasize that these were part of those public comment letters for the most part. We do not believe that laboratory developed tests are devices. They are not physical objects. They're services. We've heard some other terms related to medical activities. It's interesting to note that laboratory developed tests were not mentioned in that law that I previously described, the Medical Device Amendments of 1976. They're also not mentioned in any prior congressional hearings prior to the passage of the Medical Device Amendment. So there's no indication that Congress actually intended to give the FDA the authority over laboratory developed tests. It's not on this slide, but it's also interesting to note that it took 16 years for the FDA to publicly acknowledge that it had just learned about the existence of laboratory developed tests. So when you hear that the FDA has maintained a policy of enforcement discretion since 1976, that is not an accurate statement. They've only first acknowledged LDTs in 1992. The FDA is actually making some pretty big interpretations in their proposed rule on terms like interstate commerce and commercial distribution. I believe that Congress, when it set up the term interstate commerce, meant what it meant, commerce between states. The FDA is claiming that even if your reagents or ingredients for reagents come from another state, all of a sudden that means that your test is distributed across states. They also believe that the term commercial distribution, distributing a physical product across states, essentially means the same thing as putting an LDT on the market. So they're making some pretty broad claims here that could impact some constitutionality issues. I'll show this on the next slide. We don't believe that the FDA actually has the authority to regulate states and state-owned entities because they don't fit the definition of a person in federal law. So if you are working at an academic medical center that is owned by a state, this could be a very important and relevant issue to you. Again, it'll probably take litigation to be resolved, but the FDA may be overstating their authority here. Another interesting point is for decades, physicians and clinical laboratories have actually been exempt in federal rules from registering with the FDA, and the current proposed rule actually conflicts with that exemption without getting rid of it. So you can see some conflicting requirements coming down the road. On this states and state-owned entity issue, I won't dwell on this too much, but in June of 2020, Health and Human Services conducted their own internal legal analysis. This was ultimately leaked, and so it's freely available on the internet. But their own lawyers said that it is unlikely that FDA registration, pre-market review, and reporting requirements would apply to any state-owned laboratory, whether that's a public health or a university laboratory as well. It's interesting to note the FDA omitted any discussion of this potential significant legal limitation in their proposed rule or in their regulatory impact analysis. Now, granted, this prior legal analysis is not binding on Health and Human Services, but it is interesting that they are proposing to do something that is completely opposite of what their lawyers previously suggested that they had the authority to do. We point out many what we believe are flaws in the FDA's regulatory impact analysis. So when they released the proposed rule, they released a document that outlined their justification for the proposed rule. And there's something in the law, so this is in the law, called the general rule as part of the medical device amendments. The FDA shall not impose requirements that are unduly burdensome when they take into account the cost of complying with the requirements as well as the need to protect the public health. So they can't create rules that are so burdensome that nobody can afford to comply with them. So they are legally responsible for maintaining this balancing act, protecting the public health, but also ensuring that people can comply with the requirements that they're proposing to do. And we have concerns on both sides of the spectrum. We believe that the regulatory impact analysis basically skewed it and weighed it one way in which is not reflected by the facts. We are concerned that the FDA overestimated the purported risks of laboratory developed tests, that they ignored the clinical benefits of LDTs in entirety in the analysis, and they did not factor the negative impact from loss of essential testing services on patient care. That is a harm to public health, not a protection of public health, and that's not discussed. We're also concerned that the FDA overestimated the financial benefits to society. They underestimated the cost to patients and health systems, and the compliance costs would be prohibitive for most clinical laboratories. So this balancing act that they're required to do is actually not being conducted appropriately by the proposed rule or their analysis. Just a few quick examples. We've gone through the math on several of these things. In that regulatory impact analysis, the FDA claimed an economic benefit of the proposed rule of $39.5 billion to society. We actually went through the math and the evidence and saw several flaws along the way and have demonstrated that we believe they've made an over 250-fold error in this calculation that takes the economic quote-unquote benefit to 162 million from $39.5 billion, and therefore less than their estimates of the cost to society, which we believe those are underestimated as well. So example one, they claim that 50% of test orders are essentially LDTs. Our own data from our health system shows that that's actually less than 4% of test orders by clinicians are LDTs. FDA claims that 50% of diagnostic errors are attributable to laboratory tests. Our real-world evidence from a literature search shows that it's about 1 to 4% of diagnostic errors can be attributed to faulty diagnostic test results. Each one of these are 10-fold errors, so you can see how this starts multiplying and ultimately gets to that 250-fold error. The FDA has also claimed that 40% of LDTs are problematic, but the data they cited for this has been contradicted by a peer-reviewed reanalysis, which demonstrated excellent performance of laboratory-developed tests. So we think there's a significant problem with their source data. And our own evaluation, we're still working on this, so it's not published yet. Our retrospective analysis has demonstrated comparable proficiency testing performance of LDTs versus FDA cleared tests, which directly contradicts their belief that LDTs are problematic. We believe there's a lot of information that is also just completely omitted from their public regulatory impact analysis. It lacks any assessment of clinical laboratory financial resources to comply with the proposed rule. We also believe that most clinical labs will not be eligible for small business discounts. If they're part of larger health systems, they would exceed that revenue threshold for business discounts. So we don't think that's going to be applicable in many settings. They didn't analyze how many laboratories would have to discontinue testing due to the increased compliance costs. They didn't factor in increases in pricing due to lack of competition with labs dropping out of the LDT market. They didn't analyze the reduction in patient access to essential testing, and they just didn't analyze the negative impacts of the proposed rule on public health, which we think are very significant. Another interesting factor here is there are clear implications, at least in my mind, to future pandemic and public health response. When you think about the FDA proposed rule in the context of pandemics, it would now prohibit LDT offerings prior to a formal declaration of a public health emergency or activation of the Section 564 Emergency Use Authorization or EUA provisions. Currently, I believe you are legally allowed to offer an LDT in an urgent public health response, but it would be prohibited in this context. And I think that just dramatically slows our ability of our country to respond, not just to emerging infectious diseases, but also chemical threats and radiologic threats. And when the FDA advanced their final rule to the Office of Management and Budget earlier this month, they said this rule has no impact on pandemic response. This is a screenshot from the federal website. And so I brought that up in a call to OMB within the past week as well, because we believe that's inaccurate and not consistent with requirements for advancing a final rule. There are other things that are not included, at least in the proposed rule. There are no grandfathering provisions. These are provisions that would delay negative impacts of the proposed rule, but we don't believe grandfathering would eliminate the negative impacts. It just slows it down. And we strongly believe that labs need flexibility to maintain and update laboratory developed tests due to things that are very common in clinical labs. We've all seen these: equipment replacements, supply chain disruptions, assay improvements. All of this potentially would have to go through the FDA in the future. And you can't wait months to hear back when you've got a critical supply chain disruption. You've got to have the flexibility to move forward and validate things appropriately under CLIA. Test modifications are also something that concerns us a lot with the proposed rule. Modifications are not bad. They're often very beneficial, and they can be fairly common under CLIA for things like source types, for body fluids, for specimen containers. I'm thinking about like EDTA plasma versus citrate plasma validations, specimen stability, as well as time, temperature, and light protection. Things are modified sometimes just to automate, to keep up with the volume being sent to your lab, and that's something that potentially could require FDA oversight. The proposed rule clearly favors the concept of manual testing and believes that manual testing is safer than automated testing. As a medical director of automation at ARUP, I have a hard time saying that out loud, given the amazing improvements we've seen in testing that's automated, but that is the way that the FDA has portrayed it. And test modifications are often done, as I mentioned in the last slide, for supply chain disruptions. So we strongly believe that validated test modifications are essential to clinical practice and should remain under CLIA. We also believe there is a clear role for CMS in LDT oversight. One of the major strengths of CLIA is actually its flexibility. We've alluded a little bit to this in the last presentation as well. It allows the lab to adapt to clinical needs while still requiring validation to ensure quality. And this has worked well for decades, and they're basically proposing to throw this out the door for very rigid FDA oversight that doesn't allow for flexibility. And there's a clear public health risk in that. I strongly believe that CMS needs to be a part of the LDT regulatory conversation. And CLIAC should be empowered, not prohibited, to discuss LDT reform as part of CLIA modernization efforts. So I have some very clear concerns there in the ability of CLIAC to speak openly about these concerns. So we wanted to gather a little bit of extra information on how the clinical lab community has perceived the proposed rule. I'm going to share a little bit of data here. We did a customer survey that was distributed in February, so just early last month. And we received responses from over 500 clinical laboratorians across the country, representing 45 US states. This just shows the distribution of respondents. Many of them were in hospitals, both community hospitals and academic hospitals, as well as independent reference labs and children's hospitals as well. And the data is scary. We asked respondents about the impact of the proposed rule on their clinical laboratories, and I'll quickly go through some of the earlier responses that we got here, the initial responses in the first part of the article. Only 8% of clinical laboratorians supported the FDA's proposed rule. Almost 2/3 of clinical laboratorians who responded to our survey perform LDTs in their laboratory. And so we asked them the following questions to this subset who perform LDTs. Almost 84% of these clinical laboratorians believe their laboratory will be negatively impacted by the proposed rule. Almost 2/3 already believe they would remove tests from their menu if the proposed rule is enacted. Another third don't know yet, and that's fair because we're waiting to see the final rule. And then only 3% of respondents believe that they have the financial resources to comply with FDA user fees. So think about this data and go back in your mind to that balancing slide that I showed earlier about the cost of compliance versus benefit to society, right? The balance is off here, and I think our customer data is showing this very clearly. We then asked many additional questions about their concerns related to patient access to essential testing, financial resources to comply, impact of the proposed rule on innovation, availability of personnel resources, and the FDA's own ability to implement the proposed regulations, as well as increases in test prices and send-out costs. And the take-home point is this. In here is over 80% of respondents were either extremely concerned or very concerned about all of these. So there is a very profound concern in the clinical lab community on the impact of the proposed rule. We've released all of the survey data. There's actually a lot more than that. It's publicly released on a preprint archive. I've included the link here. If anybody wants to email me directly, I'm happy to just send them that paper or survey results directly. And you can reach out to me with any questions as well. So I am going to pause here, and I think we're going to break out into our open discussion shortly.
got it.
So we don't need to hold them.
You can pick up the handheld mics.
All right. So I will actually kick it off with a question. And of course, first I'm going to kick it to you, Jonathan, because it comes from your talk. You actually mentioned, 4% of the testing that's ordered are LDTs, right? And just sitting there, I thought about there's certain disease states where it's clearly not 4%, right? So you're thinking of all of lab testing that's being done, right? And the thought that went through my head, and maybe it's not the perfect analogy, but the keystrokes that I use to log into my bank account are 4% of the keystrokes that I'm going to use when I'm going into balance, what I'm doing, but they're still the essential 4% to get me into my account to do those next steps. So I just kind of want us to talk a little bit about the fact that it is 4%, but how important that 4% is to patients, and for flow cytometry reasons, specifically oncology patients.
Absolutely. And let me clarify a little bit on that. The 4% number came from a study we did at our academic medical center. So we looked at all test orders over the course of an entire year in the University of Utah health system, and then broke them out based on whether they were FDA cleared or approved LDTs versus emergency use authorizations, for example. There's another category of very standard things. It's a standard category we use from New York State. So that's of all test orders. You are 100% right. There are also some domains that are domains of medicine that are heavily reliant on LDTs. And so as part of that study, we also broke out the most common LDTs across all different major clinical services in our health system. And that's also in our paper as well. It is incredibly important. LDTs are incredibly important in things like hematology and oncology, particularly with flow cytometry in this group, but also molecular testing as well. So the 4% doesn't get at the true patient impact. We used it really to get at this concept of leveling the playing field. The FDA and several organizations like saying they're trying to level the playing field so that IVD manufacturers are competing on the same playing field as LDTs offered in clinical laboratories. And we are saying they already have 95% of the market. I mean, how level do you really need to get? It's really about ensuring quality of testing and not leveling any type of commercial playing field.
The one thing I wanted to add to Jonathan's comments is, even though the percentage of LDTs might be low, 4%, 3%, 2%, it aids in the final clinical diagnosis of a patient. So it's sort of like taking a bolt out of your car and putting you on the road. It's not necessarily about the percentage, but it's taking the tools away from physicians to make a decision.
Sarah, will you put that slide up that we had prepared? So I'll come to that one next. I think my next question is, and this is to anyone, why now, right? So I think we've been kind of seeing the FDA care about this topic, but why now? Why the urgency? And then it's unlike them in a proposed, ruling to, if asked to give more time for responses. And you guys might know better, but I think there were over 5,000 responses that were documented, and no additional time was given. So any idea around now, like what's the urgency and why not give a little bit more time for labs and bodies to respond? I don't know who wants to take it. Yeah.
Well, the easy answer is we don't have the person to answer that question here. So
I just didn't know if there was, if people throughout this, do you know, Jonathan?
Yeah, I'm happy to chime in a little bit here as well. And this is just my opinion too. I think there is a certain amount of this that is political, and there are changes in political perception of FDA authority and oversight that have happened between administrations over the last decade or two. I think they view this as a window to get a rule through before a new Congress or new administration as well. There's something called a Congressional Review Act. It's a law that basically says if a new rule is introduced too close to a congressional election, the next Congress can actually choose to just throw that rule out. And so there actually is a deadline. It's roughly toward the end of May that the FDA would have to introduce a new rule and avoid the risk of the Congressional Review Act. So many people in the community believe that the lack of granting extensions, which is unconscionable to me, but that lack of granting extensions may be due to their pushed schedule to try and get this out before the end of May.
So another criticism, just broadly has been that and I think you hit on it already, but the fact that there hasn't really been real world evidence, right? Anecdotal evidence is not evidence. What could have been done to make sure that there was more real world evidence of, I think, on both parts, right? Failures of IBD tests and real world evidence of LDTs being dangerous. I kind of liked how Dr. Evans put it. She said, of course, there's, bad actors out there, but you don't, suppress all speech because of the bad actors. So Virginia, what else, do you think they could have done to find real world evidence? I mean, to me, I guess when I'm sitting there thinking, I'm thinking, okay, what do we actually really end up doing in medicine? When we find a novel case, we do a case study, right? We say, hey, listen, there was this case, he presented like this, we use this test, and this is the new end result of it. And so I just kind of think, was there a deeper literature review, or is there really only anecdotal evidence against LDTs? And maybe that's the wrong question, but that's one that I had sitting there.
Maybe I'll take this one. So one of the things that I believe is the practice of medicine has evolved tremendously, not just overall, but in different parts of the country. Here in Texas Medical Center, Baylor College of Medicine, everywhere has different ways that they approach patients through the innovations that they've done. So that needs to be understood first. So we don't know. I don't know what happens here. You don't know what happens in Virginia. So that those types of medical approaches and LDTs, we need to have a better understanding of that in the context of medical care before you start putting rules into place. That's going to affect the overall care in the United States.
Anything to add, Jonathan?
Yeah, I mean, I would add this could have been a benefit of extending the public comment period. I'm just speaking for myself. I wanted to take more time and really dig through some of that anecdotal evidence that the FDA shared. Some of it is really just in footnotes as well. But you focus on what you had time and resources to focus on. And so we focused on where we did in our public comment letter, but would have loved the opportunity to dig a little bit more deeply on their evidence as well. I think there's an important role for CLIA here too. We talk about, the FDA is like saying they have no idea how many LDTs are out on the market. CLIA accreditation organizations, like CAP requires you to keep a list. New York requires you to submit LDTs to the state before they're offered publicly. That information is available. CMS even has a listing for manufacturer in CLIA applications. So you have to report your manufacturer in your application process. They're just not compiling it in a way that could help inform the public debate and then could really help be used to see what LDTs are out there, which may be riskier than others. I think the information is there. There just has not been a proactive stance from certain federal agencies on using that wisely to inform the public debate in a more transparent way.
We have a question online from Jenny Escher. It says, is it true that doctors prescribing treatments and medications based on LDTs are at risk of prescribing, quote, off-label? And is this why these providers are in support of this rule?
I would kick that either to you, Oral, or to Jonathan.
Both answer. When we practice medicine, we don't have the FDA playbook open on our office desk to decide what to give what patient. So on a given day, whether a treatment is prescribed on label or off label changes based on what the patient's needs are. Of course, we have a duty to the patient. We're regulated by medical boards on what we do. And what we do and what's in the guidelines is something that doesn't always agree with what the FDA indication for a treatment of a disease is. Just to give you a simple example, dose of an antihistamine might be beneficial for somebody with hay fever, but we might sometimes give the 4 times of that medication, the dose, to someone who's experiencing flare-up of hives. Is this necessarily an FDA-approved treatment dosing regimen? No. So do we not do that to the patient? Of course not. Jonathan?
No, I think you said that very well. I mean, it really gets to practice of medicine and the flexibility for a medical provider to do what they think is in the best interest of that patient that is in front of them, whether that be prescribing drugs or using FDA cleared approved tests versus LDTs. That is their right. I also believe there's medicine in the lab, as was described very well earlier as well. Medicine is not just outside the laboratory. Laboratory medicine and clinical pathology is a board certified medical specialty. And those physicians, clinical pathologists, should have that same medical flexibility that we grant to physicians who are outside of the laboratory. They're both practices of medicine.
Just one more thing to add to the person who asked the question. It's not, the issue is not that if the FDA, I'll use very concrete terms, bans it, that we won't be able to do it because it's banned. We just would not even have the tools.
Did you have a question, Abby? Okay.
Hello. Hi, I'm Abby Kelleher, and I work at BD. But I've also spent most of my career in the laboratory with laboratory developed tests. And I've been following this since probably 2014 as well. And one thing that I've never understood, and maybe you guys can help me understand that, is what does the FDA gain by this? And of that, you know, and I've heard the leveling the playing field piece. What would make the FDA think that it is their responsibility to level the playing field for, you know, labs and manufacturers?
Jonathan, I'll let you go first.
I'm happy to start with that. You can probably see me smiling.
Yep.
FDA it is a truly awesome responsibility to ensure safety and effectiveness of devices. And I don't want to trivialize that at all. They don't have the resources to actually get through the current number of submissions that they already have. I think the proposed rule does dramatically increase their resources, but at the expense of the clinical lab community that can't afford that. So that's where I think that balance is also certainly off. You hit the nail on the head there, though. The FDA has no authority whatsoever on the playing field of a commercial market. That has nothing to do with their statutory authority. And I've always been surprised that they've made statements about that publicly, because it is odd that a justification for a proposal is something that the agency has no authority over.
I think just something that occurs to me, right? And when you were talking, Jonathan, and you were saying, you know, they're looking at us as a manufacturer versus actually a service, right? Where you're taking this body of tests and then your clinicians are putting together a diagnosis or a prognosis based on, you know, a body of information. So I'm wondering, and this is not something maybe that could be answered, but what does it look like for them to go to certain manufacturers and say, hey, there's some tests that we feel like could be standardized. And the next, you know, step is for you, dear company, to step up and, you know, put it on your device. Because I think there's, you know, for me, there's this field of, hey, you need to stay innovative and you need to do lab developed tests. And then there's this other place where you're putting a lot of onus on a lab to reinvent the wheel every time, when there are maybe some tests that manufacturers could step in and get to an IVD status and put through a medical device. So I put 2 slides in here. This one here, I just kind of wanted to make sure the audience saw the flow cytometry is just a small portion of the LDT market that's growing. This is from 2022, but you can see just looking at the slide here that molecular diagnostics is a huge percentage of that pie, as are immunoassays, among other things. And then I threw one more figure in, the one that's right after this, and this is actually from the Immunodeficiency Society, and it's, you know, talking about what's the difference between a commercial test that has that product that's sold that has gone through the FDA versus this lab developed test that the service itself is being sold. So I'm not sure if there's more questions or comments, but I think for me, one that I want to know is, you know, what can we do, right? We're obviously not all going to individually write a response to the FDA, but what can we do to support labs like ARUP and labs like Amerimmune and, you know, efforts like standardization? What can we do to make sure that this translational medicine lab developed test bridge builder is not taken away? Or if it is taken away, that there's a response such that we can get some of those innovations back. Is it writing our senators? I mean, I don't know. It's honestly, what can we do? It's
happy
great to learn this, but what's next?
I'm happy to start here. I'm sure we're going to get some great ideas from the group. Having a relationship with your representatives and senators where they know your laboratory exists is incredibly important. If the FDA proposed rule is finalized, there's likely going to be a lot of discussion about a legislative fix that fixes what the FDA just broke. And so it's going to be incredibly important for your representatives and senators to know that you exist. Ideally, to have seen your lab, invite them on lab tours. Certainly coordinate that with your health system if you're part of a larger system. But it helps tremendously when they know you're doing good work. I think we also really have to advocate for the strengths of CLIA because CMS is not advocating for that, unfortunately. And there's incredible flexibility and power under CLIA. I think about a few things like the idea of continuous quality improvement, right? It is at the heart of all of our laboratories. We strive to make our tests better, to make our processes better, and to continually try and fix things. That is not part of the mantra of the FDA, which is about locking down a test and never changing it unless you follow a predefined change control protocol or resubmit to the FDA, right? Two very conflicting philosophies. And I'd hate for the continuous quality improvement to just get thrown out because we end up in this dynamic where things are locked down. So speaking on behalf of CLIA and the strengths of that, I think is also important.
So I 100% agree with what Jonathan has said. Last week, there was a hearing of the House Energy and Commerce, the Subcommittee on Health. And it was about the LDT ruling, and they invited witnesses. There were 5 of them from different organizations with different backgrounds. What's struck me the most is if somebody's coming to the House committee and saying, well, something may not be safe, one would imagine all the committee members having a concern with it. It wasn't like that. There was a split. There were people that were confused, congressmen and women. There were people more on the proposed FDA rule side, and there were also people that were against it. So when you see that, you ask yourself, this can't be just a safety issue. Because we all want things that are safe for us, for our families, for ourselves, and the public.
Can I add one other thing? Likely when the rule is finalized, I think another thing that's going to be really important for all of us is to understand what is in that rule and understand what it means for our labs, because our hospital administrators are going to be turning to us to help explain what the impact of this is and the timing. So now is the time to be engaged and to engage with your professional organizations who will almost certainly provide kind of simplified guides to what's in the final rule.
Yeah, that's good. So Jolene, just one to you. If there was no lab developed testing available at Harvard, what would you guys be testing for?
The same thing that we're doing now, and especially with immunotherapy coming out, like daratumumab, there would be no way for us to find plasma cells now going forward unless we had another antigen that we were able to put in, our antibody that would be able to put in our test to find those plasma cells in those rare events. So what would be the next immunotherapy, and could we find them and detect residual disease? So we would be stifled, and yes, we could do validation and show that to the FDA, but validation takes time. It took almost a year for the lyric. So that was just a whole system, but maybe for a panel that could take time, and then you have to wait for FDA to approve it now, and there's no way you can use it until they approve it. So that could be time that you have to send it out.
Yeah. So what percentage of your testing would go away?
Oh, I don't know. I mean, I don't know what
More than
immunotherapy would be.
50%?
Yeah, I don't know.
Oh, so
Yeah,
how many
depending
of
on
your,
what immunotherapy was on the horizon.
no, just like your panels. So do you use IVD panels for diagnosing like lymphoma and leukemia?
Yeah.
Or do you use the lab developed test?
Or lab developed tests for leukemia, lymphoma, PNH, MRD for BL. Yeah.
So all that would go
So
away.
if we couldn't do that panel because it's not FDA approved, then we would have to send those samples out to an FDA approved lab.
Whatever that looks like.
Yeah.
Yeah.